I think that probably the best way to start would be to talk about my mentors. I was an undergraduate with Michael Miniker, and I was a graduate student with Colin Pittendrake, and then a postdoc with Woody Hastings. So it's three of the major figures. When I was an undergraduate at the University of Texas, I was planning to go to medical school. However, I got really interested in music just at the very beginning of college, and I hadn't really had any background in that, and I realized it was a little too late to pick up any kind of instrument at that point. So since I'd always had kind of a fairly good singing voice, I decided I should maybe take voice lessons so I could take advantage of classical music. And my mother couldn't afford to pay for voice lessons, so I needed to get a job. And there had been a class that I had really enjoyed, a professor who had been very inspiring, Michael Miniker. So I went to Mike and I asked him if he had a job, and he knew who I was because I was always asking these aggressive questions. And he said, "Yeah, I've got a job for you." And so he employed me, okay, in the lab to do a lot of different things. In the end, I was doing things like taking care of sparrows in this awful environment that he had set up for the sparrows, 135 cages in darkness, and we had to feed and water them in darkness, and we'd open the cage door, and a lot of times these poor birds would be in darkness, and you'd have to reach in and change the food and water, and it was in Texas, Austin, Texas, and there were often a lot of cockroaches in the cages, and we'd be reaching in, and there'd be this white noise going, ee, the whole time, and you'd reach your arm in to change the food and water jar, and cockroaches would be running up your arms and stuff like that. So that was part of the job. I really liked Mike. He was great, and he sort of tried to feel me out in terms of what I was really interested in. At the time, I was very interested in regeneration. I was very interested in regeneration. I thought, I was going to go be a doctor and all this kind of stuff. I was thinking about spinal cord regeneration stuff, and so he got me interested in a project on earthworms. And Mike had worked on earthworms back when he was a graduate student at some point. And one of the cool things about earthworms is that you can cut out parts of their nervous system, even the brain, and it will regenerate. And he said, you could go and measure circadian rhythms in earthworms, and then once you've got that sort of established, then you can cut out parts of the brain and whatnot, see what parts you cut out will become arrhythmic, and then as the brain regenerates, you can see what part, when it regenerates, will reconstitute the rhythm, and so you'd be able to localize where the clock is in an earthworm. And I just thought that was very, very cool. And I went back and did a lot of histology to show that, yes, you could cut out the nervous system of earthworms, it would regenerate. But then when it came to the part of finding good rhythms in earthworms, that was trouble. Okay, that was very difficult. And I spent about 3 years as an undergraduate trying to find good rhythms in earthworms. I never succeeded, okay, and that's part of what I was mentioning in just a moment, but there was a time about halfway through that tenure where I thought I was getting good rhythms in earthworms, and what we had designed was an apparatus where we had a phonograph needle, okay, that was attached to a paddle. It was down in the dirt with the earthworm, okay, and so the idea is that when the earthworm moved around, it would vibrate the dirt, and that would go up to the phonograph needle, and I was starting to get some nice rhythms where you would see a daily pattern that during the daytime, the activity would go up, and it would be very active for about 20 minutes and down, and then be active like this, and that pulsatile activity would occur during the day, but during the night it was much less frequent, and this was really exciting. And I took the earthworm out of the apparatus, and then there was no activity. I put the earthworm back, and there was activity, and this was great. And so Mike said, well, make another apparatus. So I made another apparatus, and I had two worms going at the same time, and suddenly we realized that the activity pattern for the two worms was identical. That when one worm was going up and active, the other worm was too. And so something's funny here. And we started getting worried. And it turned out that what was happening was that the air conditioner on the roof of this four-story building, whenever it came on, it vibrated the whole building at a certain frequency that my apparatus was picking up that vibration. And I was just measuring the activity of the air conditioner on the top of the building. And that for whatever reason, when I had done this control, taken an earthworm out, and there wasn't any activity, that coincided with a cold spell, so there wasn't any vibration. And then when I put the earthworm on, just coincidentally, it had warmed up, and so the air conditioner came back. So I had spent quite a long time pursuing an artifact. And when it came time as a senior to decide whether I was going to go to medical school or go to graduate school, Miniker the whole time was trying to convince me to go to graduate school. He said, "I don't think you'd be a very good doctor. You know, I think you're a troublemaker, and that you probably won't really like being around sick people." Which, when I thought about it, didn't really sound that attractive either, actually. And one time I said to him, "You know, I'm not a troublemaker. Why do you think I'm a troublemaker?" And he says, "Just consider this: if you could do one experiment that would completely obliterate and abolish everything I've spent my whole career trying to establish, would you do that experiment?" I said, "Oh, absolutely." He said, "You are a troublemaker." Okay? And so anyway, so I was a senior. I had to decide what to do. I thought maybe if I applied to medical school and I didn't get in, then I'd just go to graduate school. But I got into medical school. I got into graduate school. Didn't know what to do. And I think the reason why I decided to go to graduate school was because I had failed as an undergraduate, and that I couldn't leave it there. I still was really interested in how this whole thing worked. And so I couldn't close that chapter. And so I went to graduate school. And so then I had Colin Pittendrigg as a mentor. And Colin was a very intimidating person right from the very beginning, okay? And not really very interested in what I was doing. However, he was a very bright guy, probably the brightest guy I've ever run into. He was a very imaginative, creative person, but he had a very forceful personality. And as you say, you know, the kind of concepts we had in the field at the time were different. Although I think I've just been recently thinking that in fact Pittendrigg would really probably like the way the field is right now much more than he did in the 80s and the 90s.
He was a geneticist.
Right, he was a geneticist. And a lot of people have kind of accused Colin of being anti-molecular, okay? But I think the problem was there's a couple of episodes that he told me about that I think was what led people to think he was anti-molecular. So, for example, in the 70s and even in the 80s, people would say to him, "You know, these mutants have been established in Drosophila. Why don't you just clone the gene and then you'll have the clock? Clone per and you have the clock." And that kind of simple-mindedness just drove Colin completely crazy. He knew the system was going to be much, much more complicated, okay? He was also interested in hierarchies of oscillations, and, you know, the molecular biologists at that time, they didn't want to even think about that, okay? And the truth is that both of them were right. The molecular biologists are right in that you've got to start somewhere. You've got to put your foot in the door, and the way to do that is to clone the first gene and then go from there and whatnot. But at the time, they weren't talking like that, and that simple-mindedness just drove Colin crazy. Then also, there was, at the time at Stanford University, there was Arthur Kornberg, who'd gotten the Nobel Prize for studying DNA polymerase and how DNA replication occurs. And Colin one time gave a lecture at that Kornberg came to, and Colin gave, you know, beautifully constructed, nearly impenetrable lectures, okay, for anyone who wasn't actually in with the lingo. And Kornberg came up to him after that lecture and said, "Colin, you know, this is really very interesting stuff you're doing, but I think it's going to be difficult to understand. And, you know, the way I got the Nobel Prize is I took this one protein, and I just studied everything about that one protein and what it did, and it's, you know, and that's what you've got to do with clocks." And that attitude, that just drove Pitt crazy, because while he was a reductionist in some sense, in the other sense, he felt the system was just much too complicated to even contemplate approaching it that way. Also, people have criticized him for discouraging Konopka from doing the genetic screen, which is what really got the whole molecular biology going. And Pitt was wrong to have discouraged Konopka about that. But on the other hand, Pitt had already done a number of different kinds of crosses, genetic crosses, which clearly showed that it was going to be polygenic. And Pitt just felt that trying to make a single mutation, a single genetic mutation, was not going to work. He was wrong about that, but he was completely right that it was going to be polygenic, as we've, you know, found out since then. Well, I was a failure not only as an undergraduate, I was also a failure as a graduate student. I worked for 3 years in Pitt's lab. I worked on all kinds of different things. You know, I worked on hamsters, I worked on birds, I worked on earthworms, I worked on chlamydomonas, I worked on neurospora, I worked on cockroaches, I worked on aplysia, I worked on leeches, There was probably a few, Euglena, I worked on a lot of different organisms. And I just, oh, and also halobacteria. That was the first time I got interested in bacteria, that the possibility that bacteria might have clocks. There was some papers that came out on halobacteria indicating that they were maybe light responsive. I thought that was a good system. But as it turned out, nothing worked. I've always had the problem of designing experiments that if I got this result, it'd be really exciting and great, but if you get this result, it doesn't mean anything, okay? And friends of mine like Arnold Eskin have always told me that that is not the way a scientist thinks, and that you really want to design experiments that whether you get this result or you get this result, they both mean something. But I've never been good at designing that kind of experiment. And so after 3 years, I was in trouble. It looked like there was some real possibility I would not be able to get my PhD. I was thinking about maybe becoming a technician. And I went to Woods Hole for the physiology course in 1979. And I took that course, and I realized that compared with a lot of the other people in graduate school, I was just as smart and as good as them, and maybe better than most. And so why the hell was I having so much trouble? And I realized I was trying to do things that were too difficult. At least in the context of Pitt's lab, because I was trying to do something more cellular, and Pitt's lab wasn't really the place to do that. But there were several other people there that even though Pitt was not very interested in my project, and he and I only talk about it maybe once every 2 or 3 months, and he'd usually fall asleep. Obviously would be nodding off as I tried to be telling him about the results. That despite that, I learned a tremendous amount from Pitt as a teaching assistant for him. And then, at the time, there were 3 postdocs who became famous in their own right, and they were really the people who supervised me. Terry Page, Gene Block, and Jeff Elliott, okay? And so those 3 guys were there at the time, and they were really the people who mentored me in terms of day-to-day type of experiment things. And so at the end of these 3 years, after I came back from the physiology course in Woods Hole, I thought, well, maybe, you know, the reason why I'm not doing well is I just need to change. I need to change to a simpler project. And it turned out that there was another laboratory at Hopkins Marine Station there that worked on sea urchin fertilization and development, and I'd already done some work in that laboratory in developing methods for measuring intracellular pH. And those experiments were working fine. And so I went to that professor and said, look, you know, I think I need to change labs because the stuff I'm doing on clocks is just not going anywhere. And I've got to finish off my doctoral work. And they said, well, we would be happy to have you. And so I went back to Pitt and said, I'd like to switch. And he was glad to get me off his back because I think he was getting worried that I was going to flounder. And so I switched over, and in 2 years I finished up all the work and got my thesis on sea urchin fertilization and development, okay, and the role of pH and that sort of thing. And so that was a nice little side type part of my career where I learned a lot more cell biology than I had known. Okay, but my love was still to try to figure out what was going on with clocks. And so when I looked around to look for a postdoc, Woody Hastings actually offered me a position because I'd had a little bit of experience in his lab during one summer. Okay, I remember Pittendrig finding out that I was thinking about going to Woody's lab to do a PhD, to do a postdoc. And I said, yes, that's right, Pitt. And he says, I really think that's not a very good idea. Pitt said he didn't think it was a very good idea for me to do a postdoc in Woody's lab. And I said, why is that? He said, well, you're not going to learn anything from Woody. And I said, well, that may or may not be true, but at least I can work on the system. I think the system is really interesting. And Jay Dunlap has just finished off his PhD. There was sort of biochemistry coming at that time on the system. And so I ignored Pitt's advice and went to Woody's lab. And at that time, talking about conceptual ideas of how to get the clock system, you know, some people were doing drug studies and that whatnot, but with the exception of some work that in Drosophila and Neurospora, which was just starting on genetics, there wasn't that much of a genetic emphasis. Clearly, genetics and doing mutations and analyze that has been the approach that has been the most successful at getting the clock mechanism. But in the early 80s, we didn't really know that. And we could have imagined that mutations, genetic mutations, wouldn't be no more helpful or useful than a pharmacological approach at that time. So when I went to work in Woody's lab, the approach there attracted me of taking an output pathway, which was biochemically characterized, and then trying to figure out the steps upstream, one by one, to kind of work the way into the clock. Okay, it seemed like a good idea. In the end, it hasn't been successful. Okay, but I enjoyed it. We learned a lot about the output pathway. We published a paper showing for the first time that the clock control protein of luciferase in that algae was controlled at the level of translation, that there was actually changes in the abundance of the protein. And at that time, people like Pittendrigg and other people said, "Are you sure? That seems very wasteful." And it's interesting to think that in those days, the idea of molecules being synthesized and degraded and all this turnover that we now take for granted, at that time people just didn't really believe that that was going on, that that was an incredibly wasteful thing to have happen. And that why would you want to waste all that energy? Don't you just want to kind of modify and tweak the activity and things like that? But as we now know, of course, that turnover on many different time scales is really the way in which organisms do things. And it may be energetically wasteful, but that is the way they do it. And so we published that paper on luciferase turnover. I also solved a long-standing problem in the bioluminescence field of finding and identifying the organelles that the bioluminescence came from, which was a cell biology question. It wasn't really a clock question particularly, but that was a lot of fun. I enjoyed that. And then I had to find my own position. Well, of course, I didn't really want to keep working on the same system that Woody was, because I didn't want to end up competing with it. And so even while I was in Woody's lab, he was very gracious about allowing me to start my own system. And so I started working on Chlamydomonas, which is another algae with which you could do genetics, which you can't with Goniolex, that Woody works on. But I thought that genetics would be good in Chlamydomonas, and people had started with the Neurospora Drosophila to find that genetics was being useful. And so I thought Chlamydomonas was a good system. So I characterized, looking at phototaxis rhythms. Victor Bruce, who had worked on phototaxis and Chlamydomonas, gave me his apparatus because he'd retired from Princeton at the time, and I modified it and that sort of thing. And so when I left to go to Vanderbilt, I was already working on Chlamydomonas. But while I was still in Woody's lab, there was an opportunity to go to Japan for an international meeting of cell biology. Okay, and I thought, wow, this would be great. I always wanted to go to Japan, but I thought this is going to be my one opportunity to go to Japan in my whole life, and I really should take advantage of this opportunity. And Woody supported me by giving me some of the travel monies to do that. And so I set up to go to Japan and also take a brief one-week tour before the meeting. So I went to Japan. I learned a little bit of Japanese before I went. And on the tour through Japan, it also went fairly close to a town called Okazaki, which is near Nagoya, which is where the laboratory of Nakashima was. And Nakashima was working on Neurospora clocks. And I decided to drop in on Nakashima because I had met him when he was still in California. And so I dropped in. And it happened that Takao Kondo was also working at the time in the same place. Now, Takao Kondo wasn't actually there at the time that I visited, and this was 1984, that's right. But Nakashima showed me some of Kondo's data that he'd gotten with Chlamydomonas phototaxis rhythms, and they were actually very nice rhythms. I thought, wow, maybe the way that Kondo-san is measuring those rhythms is something that it would be great to kind of learn how to do that and collaborate with him. Okay, so when I went back to Woody's lab at Harvard, I said, Woody, we should really invite this guy, who I didn't meet, but to invite him to come to the United States and try to see about how he's doing phototaxis and Chlamydomonas. And Woody said, oh, okay, so we invited Kondo-san. He came for 3 months while I was still there at Harvard. We got to know each other quite well. We were working on Chlamydomonas. The next year I went to his laboratory for 3 months, and we did action spectroscopy with Chlamydomonas. And anyway, that established my relationship with Kondo-san and long friendship that's been going on, and collaboration for many years. And so in the late 80s, we were working on Chlamydomonas phototaxis rhythms and that sort of thing. And I went back and forth between Japan a number of times, collaborate with a number of other people in Japan. So I collaborated with a lot of Japanese in the late 80s and early 90s. Kondo-san came to my lab, I think in 1991 or 1991 or something like that. Let me think about that. yeah, about that time, to do a sabbatical. And when he came to do the sabbatical, he said, you know, I think that it might be good to look for another model system that might be easier to do genetics on than Chlamydomonas, because at the time, Chlamydomonas, you could do classical genetics, but it was very hard to kind of clone genes and whatnot. And another geneticist had impressed upon Kondo-san that yeast and E. coli were a lot easier to manipulate genetically. So Kondo came to my lab on this sabbatical and was trying to look for circadian rhythms in yeast and E. coli. And he didn't make any progress on that. One of the things he was doing, which was clever, was to see whether there might be a rhythm of resistance or sensitivity to ultraviolet light. So he was pulsing with ultraviolet light at different times of the day and night. But that didn't actually go anywhere. And so after about 4 or 5 months, when he had been in my lab on sabbatical, I went to the cell biology meeting. Okay? And I gave a poster on Chlamydomonas circadian rhythms, and right next to my poster was a poster from a group in Taiwan that was looking at nitrogen-fixing cyanobacteria and had what appeared to be a nice circadian rhythm. And I thought, wow, now that's maybe this is. So I went back to my laboratory, said, Kondo-san, instead of screwing around with E. coli and yeast, let's look at cyanobacteria. And he called up one of his friends back in Japan to ask them about whether cyanobacteria would be a genetically manipulable system, and they told him that yes, it probably would. So he said, okay, and so I requested the strain from the Taiwanese group, okay, and our plan at that time was to take that cyanobacterium, okay, it has nitrogen fixation, to clone out from that the nitrogenase gene and its promoter, try to take that promoter, attach it to luciferase, somehow transform it back into that bacterium and make a luciferase reporter cyanobacterium, okay? However, there were a lot of sort of questions about how you really do that, because this was a strain from the wild. There weren't any genetic techniques that had been worked out. It's not clear whether it was or could be transformable. That was a long road to hoe, okay? But that was what we were thinking about doing. So we got that strain from Taiwan. And then about a month later, I was visiting a friend of mine in New York City, who's just a good friend of mine. And I decided, you know, Steve Kay is here in New York City doing a postdoc in Nam Haichu's lab. I should go by and just say hello. So I gave him a call, and they said, sure, come on by. So I went over to the Rockefeller. And Steve Kay was there, and that was when Andrew Miller was also working there with Steve, ostensibly with Nam Haichu, but really Steve and Andrew were working together. And so I went in, and we were just talking about what we were doing and whatnot, and it turned out that they had the same damn idea to take that Taiwanese cyanobacterium and do exactly the same thing that Kondo-san and I were thinking about doing, okay? And they had already gotten the strain, and I was totally bummed out. Okay, because they were good molecular geneticists, and Kondo-san and I at that time were not at all. I knew there was no way in hell that we were going to be able to get ahead of them on that same project. So this illustrates something that Ephraim Racker said in his book, okay, about science, and that is that sometimes troubles are good for you, because either you learn something that you really weren't looking for at the beginning, or it'll send you in a new direction. So I came back from the visit to New York and told Kondo-san, you know, Steve Kay and Andrew Miller are just going to eat our lunch on this. There's just no way. And I said, you know, maybe what we ought to do is, instead of working on this Taiwanese nitrogen-fixing cyanobacteria and whatnot, I mean, they basically got the high road on that. Maybe what we should do is just get some other cyanobacterium that at least we know is genetically manipulatable, but that we don't know what kind of rhythm to look at in it, and start working on that. And, you know, Kay and Miller are probably going to forge way ahead of us on this, but maybe we can still make contributions. So Kondo-san said, well, okay, well, what rhythm is going to look for? And at that time, I was doing a lot of 2D gel electrophoresis in Chlamydomonas, looking for rhythmic proteins. I said, well, we can try to do the same thing in cyanobacteria, look for rhythmic proteins, and then try to identify that protein, and then maybe do the same kind of an idea, but we're going to have to find a protein first, and we don't have any candidate protein. And Kondo-san said, well, okay, fine. So I decided I needed to start calling around to other cyanobacteriologists to find out who knew about how to prepare proteins from cyanobacteria that would electrophrase well in 2D gels, which is not trivial. And as I called from one cyanobacteriologist to the next, eventually I got routed to Susan Golden at Texas A&M University. So I called up Susan and told her what we wanted to do. And she said, sure, well, I mean, this is the way to prepare cells optimally for 2D gel electrophoresis. But, you know, I just want to mention that I have a postdoc in my lab who's been looking at the levels of the PSBA1 messenger RNA, and he's been taking time points over 2 days, and it looks like it might be rhythmic. And I said, well, that's really interesting. Can you send me the data? She said, sure, sure. And, oh, and by the way, you know, we're also making a luciferase reporter of that promoter because we're interested to see whether it gets induced by high versus low light. She was interested in light regulation of gene expression in cyanobacteria at the time. And I said, oh, well, gosh, if you can send me that report strain which you made, you know, I know how to look at bioluminescence rhythms because of my experience in Woody Hastings' lab of looking at bioluminescence rhythms in Goniolex. And so she said, okay. Well, I had to keep bugging her about the data for the messenger RNA, but about 2 months later, she sent me the data, and as you might imagine, coming from a lab that didn't work on circadian rhythms, there would be like 16 hours where they'd have time points, and then there'd be like 8 to 10 hours where there'd be no data, where the person went home to go to sleep, and then there'd be the next day there'd be time points, and so it wasn't really very persuasive, as you can imagine. But anyway, there was a slight suggestion of some kind of a rhythm, but in any case, she finally did send us the reporter strain as well. And at that time, I didn't have a luminescence machine to measure that, but at that time, Takao Kondo was just about to return to Japan, and he decided he would go visit Woody for a few days on his way back to Japan. So he took the reporter strain with him to Woody's lab. Woody's lab had the apparatus for measuring bioluminescence, you know, at that time, the only one in the world to measure bioluminescence automatically. And Kondo-san put the cyanobacteria into this machine, and got data that was totally unimpressive, okay? There was like this barest suggestion of some kind of an oscillation, okay? But Woody also knew about bacterial luciferase, it was bacterial luciferase, and he made some suggestions to Kondo-san about how to present substrate and things like that. And Kondo-san went back to Japan and made a homemade apparatus, took Woody's suggestions about how to administer the substrate, which is a vapor, it's an aldehyde decanal, and started getting rhythms. And so that's how that whole thing got started. And, you know, I had to thank Steve Kay and Andrew Miller for scaring the hell out of me and scaring me away from working on the Taiwanese cyanobacterium, because that would have been a long road to hoe if I had really had to develop it from that system. But finally getting us to get in contact with Susan Golden, and then the synergy of those things just clicking together is what got that system going. So in any case, it's really the trip to Japan and then the connection with Kondo and all those things of building up that established my connection with the Japanese, and then also the cyanobacteria work. Okay, and then not long after Kondo left my lab on that sabbatical in 1991, I met my wife, who's Japanese, and so I married a Japanese, and so that established and cemented my relationship with the Japanese in another kind of a way. And so now I've probably been to Japan like 15 times, okay, and so it's really funny for me to think that back in 1984 when I went off to that meeting, I thought, this is the one time in my life I'm ever going to go to Japan. I really need to take a tour of the country to see what it's going to be like. It's so funny because, in fact, that's not the way it worked out at all. And, of course, it never would have been predictable that would have had happened. So, yes?
But weren't you going against, to look for rhythms in bacteria? This was against the...
Right. But I think in that sense, when Mike Miniker told me I was a troublemaker, he was right, okay? And that I had been thinking about bacteria ever since I was a graduate student when I first got interested in the halobacteria, these archaebacteria. And I think that when I learned about those archaebacteria that will pump protons in the light, then I thought, well, heck, if they pump protons in the light, if they're exposed to a light-dark cycle, that means they're going to be pumping protons, their pH inside the cell is going to change on a daily cycle, and you would think that that kind of an organism would evolve some kind of a clock to anticipate those changes and to make better use of those proton fluxes. It may interest you to know that I have tried about 4 or 5 times in my career, fairly seriously, to again try to find rhythms in Archaebacteria and Halobacteria, because it's a very interesting group evolutionarily, and every time I fail to find rhythms in Halobacteria or in any Archaebacterium. But I'm going to keep trying because I think it's a very important group. But in retrospect, of course, the cyanobacteria are the same kind of principle. That is, that for a photosynthetic organism, light is their source of energy and life, and that the ability to be able to adapt to that optimally by having some kind of a timing mechanism makes perfect sense. Okay, so while I hadn't thought of cyanobacteria at first, when cyanobacteria kind of came along later and already kind of primed by the thinking that I had already had about the Halobacteria as a graduate student, when I saw the cyanobacteria later on as a possibility, that's when I thought, yes, this is a very interesting system. Now, so that was my thought. I was interested in it because it challenged the dogma. And by challenging that dogma, we were kind of expanding our thinking about what was possible, okay? Kondo-san's interest, at least if I accurately can gauge it, were more practical. He was kind of responding to people saying, look, if you really want to analyze this problem, that having genetics would be good, and to do genetics on the scale that people were doing bacterial and yeast genetics, and to look for that kind of a system was a good idea. So that's, I think, what led him, once the bioluminescence rhythm had been established, and once the fact that the fundamental properties of circadian clocks were found in the cyanobacteria, that's what led him then, in collaboration with Masahiro Ishiura, okay, that was one of the people he'd been talking to about this kind of thing, of using the bioluminescence rhythm as a screen for mutants. And so using the bioluminescence, plating them out as single colonies, which is what people always did with yeast and E. coli in terms of doing a mutant screen, and using the light of the bioluminescence, okay, to look at the rhythm. And Kondo-san has always been really clever about designing apparatuses, writing software to do that. And so, he got cameras and attached them to computers and figured out how to do that whole thing. And so that was the first screening apparatus that was designed to look at bioluminescence rhythms. So it was his kind of practicality and those skills that he had, with, I think, input from Ishiura of the idea of pushing him to say, let's try to figure out a way of screening colonies on agar the way people do with bacteria and yeast. So the conjunction of those things then led to the ability of screening, finding mutants, and then basically the whole thing kind of kept rolling along in terms of cloning the genes and whatnot. So.
And he's become more and more biochemical.
He's become more and more biochemical. I've become more and more biochemical, although I'd had some experience with that in Woody's lab. Both of us have become more knowledgeable about molecular genetics. I mean, neither one of us really started as geneticists. Susan Golden was a geneticist, molecular geneticist from the beginning. So was Masahiro Ishiura. He was a molecular geneticist from the beginning. So at the time, Kondo and my kind of background and expertise in circadian clocks and screening things, I mean, looking at rhythms, and in my case, a little more biochemistry and whatnot, our expertises were similar. And then Susan and Masahiro's had kind of similar expertises. And in the end, that's led what was a four-way collaboration to break up, because the best kind of a collaboration for going long-term is where people have different expertise, that they can keep working together, and they both are helping each other, but neither one of them is kind of impinging upon the other one's territory, okay? And so even though I'm still a very good friend of Takao Kondo, in time, as we started developing groups of our own people, okay, we have to find projects for the people in our labs, and that there's only so many different ideas. And for a while, we were able to hold it together by saying, okay, well, with this paper, we'll let one of the people from your lab be the first author, and then the PI from the other lab will be the last author, and next time we'll switch it around. And for a while, that worked. But as our group started to grow larger, there became a time where that just didn't really work, okay? And again, Susan and Masahiro had sort of similar techniques and expertise, and so they always wanted to do the same kind of things. And Takao and I also tended to have a lot of the similar kind of ideas. So there were stresses that ultimately led us to separate and go our different ways, although we'll still kind of collaborate, and we tend to keep each other informed about what we're doing in advance so that we try to minimize overlap. And as time has gone on, there was kind of a bottleneck where once the genes were being cloned and just cloned, there was just a set of obvious experiments that everybody wanted to do, and that's where the strain was the worst, okay? Now, we've tended to diverge, although I'm not saying that there's still not difficulties, but we've tended to diverge where Susan and I have done more structural biology, and that's not something that Kondo was initially interested in. He has become very biochemical, and that's good. Then another thing that was kind of another kind of side project, started out as a side project, was looking at the adaptive significance of the clock in cyanobacteria, which is something that I hit upon because one time when we were interviewing for a position for an evolutionary biologist in the department, I was talking to this job candidate, and I don't know exactly how it got started, but the job candidate said, well, how has it been established that clocks really are adaptive? And I said, well, there's the anticipation idea and blah, 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 and there's all the different things. But frankly, we don't really know. There haven't really been strict tests of that. And I said to him, how would how would a population biologist test adaptive significance? And he said, well, a lot of times what we'll do is a competition type of an experiment where you might have like a northern latitude and a southern latitude organism and put them in different places and see who is reproductively most fit and just see what the parameters are that enhance fitness in a certain environment. And so I said, well, that's kind of a cool experiment. I mean, we've got these different mutant strains that have different periods. We could put them in different environments and see which one won out. And in my initial concept of that was I was going to take 5 different strains and put them in a test tube all at one time and put them under different light dark cycles and see which one of the 5 would win out. Well, it turned out that the sensitivity of the method at that time wasn't really sufficient to do 5 strains simultaneously. But what we hit on was basically just doing competitions between 2 strains, pairwise competitions, with short period mutants, long period mutants, wild type, under light dark cycles that were short and long. And that turned out to be very cool. And one of the, at least up to that time, one of the most rigorous kinds of tests of adaptive significance. We still don't really know how it works, and I'm still really interested in trying to figure that out. And maybe we'll do some mutant screens to see what it is, but there appears to be some kind of interaction between the strains. Okay, that's at least our best guess, that there's interaction so that one is actually inhibiting the other. And we're trying to figure out what the factors, whether that's through some kind of a diffusible factor or whether that's by contact of the cells. And in bacteria, there's a lot of evidence for communication between cells, either by direct contact or by the secretion of diffusible factors. So that's certainly not unheard of. It's just that we don't exactly know what's going on in the cyanobacteria to accomplish that. So I was saying that I think that Pitt would really like to be cognizant of what was going on in the field right now, because now that these people are all talking about systems biology as though this is something new and different and whatnot, that's really what Pitt was talking about, that the super reductionist idea that there's going to be like this one gene or 2 genes and that was doing everything, that was just anathema to him. But the idea of a very complicated network of genes interacting in multiple loops and whatnot, he would have been completely at home and comfortable with that. And I think that's where we've come to, okay? And that in some sense it's a vindication of what he was trying to say all along, and that the molecular biologists at the time were just, for technical reasons, they were simple-minded. That's where you have to start. Pitt really couldn't appreciate that, okay? But that where we've come to is now a stage where Pitt would feel, I think, completely comfortable and would be very excited by what's going on in the field.